Good afternoon, ladies and gentlemen. I'm Eric Ho from University Technology Petronas in Malaysia, with the honor of addressing you yet again. Deep neural networks, artificial intelligence, has received plenty of attention and excitement in recent years with the promise of revolutionizing computational analysis and of complex data sets like images, especially in the advent of big data. In my presentation this afternoon, I will attempt to distill the landmark achievements of deep neural network artificial intelligence before focusing on a very special problem of neuroimaging analysis, that is, of the high dimensionality of the neuroimaging datasets. Comparisons between neuroimaging datasets is crucial towards our understanding of how the brain evolves in under neurodevelopment, neurodegeneration, and neurodisease. And with such high dimensionality, it is not easy to arrive at statistically significant conclusions. And it is my hope to propose a preliminary method and some preliminary experiments uh, describing the work on how we are trying to achieve this goal. First, let us begin by understanding what is a deep neural network AI. A deep neural network AI is a pattern recognition or decision making system comprised which arrives at this decision through the interaction of many basic units called the perceptron. And the perceptron basically borrows from the biological neuron, where inputs from either the images or from a previous neuron are weighted through multiplication, and this mimics the synaptic weights of past neurons, and then they are subsequently integrated, just like in the cell body, and undergo a thresholding function, which models the voltage threshold required to trigger an action potential in axons. Mathematically, this perceptron model describes a decision boundary which literally separates images with features in one side of the boundary from images with features on the other side. The word deep actually comes from the interconnection, the way that these perceptrons are interconnected, not just within a layer, but there are many layers that stack one after another, ranging from tens to sometimes hundreds to thousands. And the real advance in the past decade has been the discovery of a training procedure which allows us to modulate and set the ideal synaptic weights to achieve the best classification or decision making operation. And this procedure is nothing other else but an optimization algorithm, which is iteratively performed. This algorithm is called gradient descent to minimize a cost function, typically called the cross entropy. The first major achievement of deep learning emerged in 2015 when Microsoft declared that the deep learning system, specifically the residual convolutional neural network, whose architecture is shown here, was able to achieve a decision making error far below the average human assessor in a competition of 14 million images, which contain roughly 21,000 different image classes. So you can see the architecture that was used here, where each colored box here comprises of a layer of densely connected neurons this way. And the major advance in deep neural networks here is that using a standard architecture like this, and with the help of plenty of good labeled data, one could develop an algorithmic system to make decision making through a simple training step without needing to even fully understand the internal workings of such a system. And this really opened up the um, the application of deep learning to many different fields. And the key advance here is that this process of having deep layers of interconnected neurons with a learning process that's able to set these ways automatically solved a very difficult problem in traditional machine vision, that is trying to find the best way to represent features from a data set for decision making. The second advance came from a field called segmentation, where deep learning systems were first shown to be able to segment out cells from DIC microscopy, like here, and be able to identify individual cells, and later, when applied to more complex and varied images, being able to label individual pixels as belonging to certain classes, whether they belong to a road, a car, sky, or trees. And this really was a achievement which demonstrated the ability of deep neural networks to comprehend an image and to understand the scenes in the image, because the deep network is able to provide a label on specific locations within images. And the third and most recent advance is that deep neural networks have been able to generalize beyond just being able to comprehend and locate objects or concepts within an image. The neural networks are now able to generate new instances of these concepts that they've never seen before. So the first demonstrations was the generation of bedroom images here, and these bedroom images are purely synthetic and they are not real. And of course, the faces within the middle here are all truly synthetic, being a fusion of the pair of faces here on the side here. And this was achieved by a training algorithm known as degenerative adversarial networks. So as you can see, these three major advances has led to an explosion of applications of deep neural networks, both in all the steps of MRI image analysis as well as brain analysis, from improving the acquisition, the reconstruction and restoration, as well as registration and super resolution, to decision making by identifying disease segments in the brain or classifying diseases. Encourage you to read this paper to get a broad understanding of the various creative ways that deep neural networks have been applied. But I'd like to caution that amid all this excitement and broad range of applications that are being discovered, there is a difficulty that remains unsolved, and that is, for all the power and ability of deep neural networks to accurately classify and make decisions, human beings do not understand yet how these deep neural networks make these decisions, or what hidden biases or errors may reside in these decision making systems. And this is, of course, a clear concern in the medical field. And that's why there is a push in deep neural networks now towards what is known as explainable AI, that is, deep neural network systems whose decision making process is explainable or understandable to human beings. Now I'd like to shift gears to focus on a very special problem, that is the problem of high dimensionality in neuroimaging. And I first like to bring to your attention the observation that no single neuroimaging modality is able today to give us a complete picture of the structure and functional relationship of the human brain. We have various kinds of imaging modalities like MRI, diffusion imaging, PET imaging, which studies metabolic activity, blood time of flight MRI imaging, which can give us the blood vessel distribution, EEG, MEG, gene expression networks, as well as functional connectivity networks. Each of these imaging modalities yields different types of data, and there are multiple measurements of data which are spread across space, that is over the entire brain, surface on volume, and also across time in the form of synchronization or interactivity between different brain points. And besides the multiple data in space and time, we are also extremely interested in the connection between how these data points in space and time are related, and how they interact with each other, not just within a single modality, but cross modality, which is relationships that have still not been explored today. And the difficulty with such exploration brings us to the problem of multiple comparisons in statistical hypothesis testing. To explain the multiple comparisons problem, I like to take the example of a single three-dimensional structural MRI brain image, say with 100 voxels in X, Y, and Z direction. Such a brain image has easily 100,000 voxels, where each voxel has a unique measurement of either brain volume or cortical thickness. If we intend to make a decision with statistical significance of p-value less than 0.05, this means that we are 95% confident that our decision will hold true for 95% of all the images that we see, or all the voxels that we see. This easily means that within one single image, we will have random chance occurrences of apparently significant differences which are not real, they are just statistical artifacts in over 5,000 voxels. So this is really a problem because the sheer number of comparisons, that is comparisons on individual time samples or space samples, lead us to a lot of random chance random decisions that we can say nothing about statistically. Our conventional hypothesis testing mechanism argues that the only way to overcome this problem is to get a large enough sample size with sufficient diversity covering all possible occurrences in the human population. And there are techniques, namely the Bonferroni correction or familiarized error corrections, that allow us to manage the multiple comparisons problem. But these statistical techniques end up only giving us conservative estimates, and this means that very subtle differences, which could be significant, but because we do not have sufficient sample size, will never be detected in our data sets. And so we miss out on the subtle differences, which we are as yet unsure could be meaningful or not. Two alternative techniques. One is the Bayesian hypothesis testing, also fail to satisfactorily handle the multiple comparisons problem because Bayesian hypothesis testing needs accurate prior probability estimates, and the prior probability estimates is an estimation of the distribution of the independent covariates of factors in the population, that is the distribution of age or cognitive scores, and this is something that cannot be known with great accuracy. More recently, permutation testing or bootstrap methods, that is methods that try to randomly shuffle the known measurements to see how likely or unlikely is the specific set of data in that particular appearance order, how likely or unlikely that is to arrive at statistical significance. So this technique, although achieving some good results, still is a little bit simplistic because the idea here is that even in a cohort of, let's say, 1,000 MRI images or 10,000 MRI images, we may still be undersampling some aspects of the population, and that in these undersampled data spaces, the permutation testing and bootstrap methods are making overly simplistic assumptions. Therefore, I wish to propose a novel framework to utilize deep generative neural networks as a method for comparing high-dimensional pathologies in the high-dimensional neuroimaging images. So the idea is simple. Instead of comparing individual brain images with measurements at individual space and time points, I propose that we should use deep neural networks to create statistical models of the data set. So multiple statistical models of data set, each with slightly different assumptions to complete the undersampled space, and we should instead compare the models for statistical testing instead of the individual brain images. And this is a difficult and challenging mathematical problem because first we have to ensure that we have our deep neural network models can sufficiently model the complexity and the coverage of features or brain features present in the training data set, and that where these models try to make guesses or generalize in undersampled or unsampled features in the population, these generalizations should be physiologically accurate and feasible, and that we need a fair mathematical measure to compare the similarity of differences between models. So this is a challenging mathematical problem. And instead of solving this problem, we try a preliminary experiment to solve a simple problem that is addressing the high dimensionality in structural MRI T1 anatomical images, that is images with volume, thickness and shape variations. So these images have high dimensionality because each voxel has a measure of volume or thickness, and frequently we have a lack of sufficient data set size and coverage in order to train good classifier. So I will next go through some preliminary work we have done trying to use deep neural networks to invent new ways to accurately model the brain structure and to identify healthy brains, whether brains are healthy or not, brains that the model has not previously seen before. Our approach is to use a generative adversarial network algorithm to train two deep networks. They are known as the discriminator with this architecture and the generator here. The role of the discriminator is to decide if a particular MRI image is really similar to the training data set or different. And the role of the generator is to synthesize this perfectly synthetic three-dimensional T1 anatomical MRI image. And instead of training our deep neural networks on the raw images, we warp we deform individual images to an MNI 152 template and train on the deformation in X, Y, and Z instead. And then our generator would create new instances of deformation which match the statistical properties of the training data set, and then deform the standard break back to generate synthetic images. And the guiding idea behind this approach is to get the shape and the sizes of brains that we generate correct before solving the problem of trying to vary the tissue type. So as the first step of visualization, I'd like to show you the results of axial slices of synthesized or fake brains. This is one synthesized brain, another synthesized, and a third desynthesized brain. And here you see an illustration of the deformation field. And as a point of visual comparison, these are the same slices at a similar MNI location for different individuals. So our synthesized brains look sort of realistic. We next ask the question, do the deformations in the synthesized brain match the deformations in the training data set, that is the real brains, accurately? And here I plot the statistical distributions. They approximate a Gaussian distribution of the deformations in X, Y, Z, and radial deformations of the five major brain lobes, the frontal, parietal, temporal, occipital, and cerebellar lobes. And you can see the blue curves approximate the training real data set, and the red curves approximate our synthetic data set. And there is somewhat good approximation, a somewhat good match. As a third step, we try to assess a finer measure of accuracy, that is, how similar are the shapes of specific brain regions to between our synthetic data set and real data sets. For the whole cortex, the similarity is very good because the p-value is very high. It's not statistically significant, so we cannot differentiate between real and synthetic brain data sets. But we still have a ways to go with a few structures such as the cerebellum and the thalamus, where there is an inaccuracy or mismatch between shapes. So why is these deep neural network that I've presented to you, how could it be useful? Well, I'll suggest that the discriminator aspect after the training process is able to discriminate between real and synthetic. Is able to give a decision on whether data sets, whether any new image data sets match the training data set, and this can function as a classifier, decision-making unit to determine if brains are unhealthy or anomalous, and we can furthermore explore for brains which have anomaly, the nature of the shape deformations through the shape descriptor. And we are proposing to extend this further work in future to investigate how to include diversity in the folds and the gyri and sulci of the brain surfaces to improve the region-specific classification, first by improving regional shape accuracy and improving the intensity values in the MRI, and finally to look into developing new methods to compare global and local structure relationships. And I'd like to conclude by extending an open invitation for any of the clinicians or scientists here. If you have a clinical application and MRI data that you think might be a good fit to our proposed model and approach, please feel free to reach out and contact me at the email here. I thank you for your attention.